Hi, this is Ms. Rahman, and we're going to do a walkthrough of the protein folding lab. This is one of my favorite labs. It's a lot of fun. It's like solving a puzzle. The instructions to build your protein are in Schoology. You're going to follow some rules on the folding of the proteins. The amino acids with the white clips are hydrophilic, so they're going to point outwards towards what would be the cytoplasm of the cell, which is aqueous. So these amino acids will hydrogen bond with water. And even if you have to just turn the clip to the side to make it facing out, that's completely fine. Everybody's protein will end up looking different from each other because you have randomly added your amino acids. So just make sure that they're pointing out. In the pictures you're seeing, this is a protein that I folded, and it's all one structure. I've just turned it around so you can see different aspects. Here you see the acidic and basic amino acid side chains. They're also facing outwards towards the cytoplasm, but then they're also facing each other because there is that electrostatic interaction between the positive and the negative charges. And once again, even if you just have to turn the clips to the side, that still counts. You can move those clips around 360 degrees if you need to. Next, we're going to see all of these together. So you see the hydrophilic amino acid pointing out. Then we have our acidic and basic amino acids facing outwards also, but facing each other because of that electrostatic interaction. Now the hydrophobic amino acids, they have a yellow clip. And this is probably going to be the most difficult part of the lab, is trying to get them in the center of your protein. So notice my protein is very compact and it is bending every single direction. So don't be afraid to fold up your protein as small as possible. It's going to look really weird, but that's the point, is the protein will fold so that it meets all these requirements. And once again, even if you just have to turn the clip around, that counts. The green amino acids are the cysteine residues, and they form that disulfide bond. The disulfide bond is a covalent bond. It will be hydrophobic when it forms, and it's going to be on the interior of the protein with the hydrophobic amino acids. Here's my final protein showing all of the different interactions at one time. You can see the disulfide bond is in the very, very back. It's hard to see. And the hydrophobic ones are as well. But that's sort of the point. They need to be on the inside and difficult to see. All right, if you have any questions, just let me know.